This video will discuss on mRNA translation. At the end of this video, students should be able to describe the process of mRNA translation. According to dictionary, to translate means to transfer or turn from one set of symbols into another. Therefore, mRNA translation refers to the process of turning genetic information stored in mRNA into protein. mRNA will be translated by ribosomes in the cytoplasm. Many ribosomes will attach to and translate one single mRNA into protein simultaneously. This observation is called as polysomes. In prokaryote, polysomes may form as the mRNA is being transcribed from DNA, since there is no nuclear membrane separating chromosome from cytoplasm. In eukaryote, polysomes form in the cytoplasm after the mRNA has been transported out of the nucleus. Polysomes help in speeding up protein synthesis. They look like beads on string under the microscope. During mRNA translation, transfer RNA will carry amino acids for protein synthesis. tRNA has a clover-like structure. Each tRNA has an anticodon site that is complementary to the mRNA codon. tRNA carries specific amino acid depending on its anticodon and delivers it to the growing polypeptide in ribosome. Next, we will learn about mRNA translation process in eukaryote. This process is very similar to mRNA translation in prokaryotes. It involves three phases of initiation, elongation, and termination. During initiation phase of mRNA translation, there will be formation of pre-initiation complex, activation of mRNA, as well as formation of initiation complex. First, the 80S eukaryotic ribosome will separate into two subunits, one large, 60S, and another one small, 40S. The initiator tRNA that carries methionine, or mTRNA, will combine with eukaryotic initiation factor 2 or EIF2 that bound GTP. Together, mTRNA, EIF2, and GTP associates with the small ribosomal subunit, the 40S addition of other eukaryotic initiation factors will form the first pre-initiation complex. Next, mRNA activation will take place. A group of EIF4 proteins will bind to the 5' cap of mRNA to straighten the mRNA molecule. EIF4 proteins will also assist ribosome in scanning for the start codon. The first pre-initiation complex will recognize mRNA via EIF4 proteins attached to the cap and scans for the start codon. This process requires energy in the form of ATP. The correct alignment between start codon AUG and mTRNA anticodon marks the formation of second pre-initiation complex. Subsequently, large ribosomal subunit 60S will reassemble with the second pre-initiation complex. Energy from GTP will be used up during this reassembly. The eukaryotic initiation factors as well as GDP will be released, and the initiation complex is formed. Within the ribosome of the initiation complex, there are three spaces for tRNA attachment known as the acceptor or A site, peptidyl or P site, and exit or E site. Now that the initiation phase is complete, the elongation phase will take place. The next tRNA carrying an amino acid with anticodon matching the next codon on mRNA after AUG will bind to elongation factor 1. The EF1 also carries a GTP, similar with EIF2. Energy from GTP will be used so that this aminoacyl tRNA could enter the A site and bind to the mRNA codon. Once energy in GTP is used, EF1 and GDP will be released. The amino group of amino acid attached to the tRNA in a site will attack the carboxyl group of methionine in the P site. This reaction will be catalyzed by peptidyl transferase activity of the ribosome. Thereafter, methionine will get transferred and attached to amino acid of tRNA in A site. Meanwhile, tRNA in P site is now deacetylated. It does not carry amino acid anymore. Anticodon of deacylated tRNA will still attach to the mRNA codon, but its other end will enter the E site. Then, the deacylated tRNA will leave the ribosome. Subsequently, elongation factor 2 or EF2 carrying a GTP will bind to the aminoacyl tRNA in A site. The energy in GTP will be used, and the EF2 as well as GDP will be released from the ribosome. This will trigger the transfer of aminoacyl tRNA from A site to P site. The A site is now vacant for the next aminoacyl tRNA. The mRNA moves along as well. The elongation phase will occur repeatedly until a long polypeptide chain is produced. At the end of elongation phase, stop codon will be reached. No tRNA has anticodon capable to recognize the stop codon. However, this stop codon can be recognized by releasing factors. Releasing factors that carry a GTP molecule will enter the A site and bind to the stop codon. With the help of peptidyl transferase activity of ribosome, releasing factors will break the bond between the growing polypeptide chain and tRNA via hydrolysis. This hydrolysis process requires water and energy from GTP. The polypeptide chain is then released. After polypeptide chain is cleaved, the whole complex disassembles. mRNA, deacylated tRNA, releasing factors, and GDP are all released. 80s ribosome will dissociate into one large, 60s, and one small, 40s, subunits again, ready for the next translation cycle. This is the end of this video. We will move to the next learning activity, which is self-directed learning, or SDL. Students are required to find out the answers to the given questions in this SDL session and submit it to the lecturer in charge. The deadline for submission is 2 weeks from today. Should you have any inquiries, please contact Dr. Khairul Anwar Zarkasi at khairul.anwar@upnm.edu.my. Your submission will be graded. The marks given will contribute to the 30% of continuous assessment in this module. Thank you for your participation.